Well, hey, AP Biology, hope you're doing well when you watch this video. So this is, I think, is part 6. We only have 2 more video notes to do before we kind of review in class and finish up really well, really strong. We're going to do our best, all right? Not sure why there's a flower on the... Oh, yeah, someone just drew that right before class ended. That's a pretty good flower. Anyways, yeah, let's go ahead and get started, okay? So this is part 6, and this is all about phylogenetic trees and how you can see that there's, trees, a lot of different trees over here and various things. And so it's, A, B, C, D, E, we're going to talk about common ancestry and roots and all that sort of stuff, okay? So let's dive into some of the specifics of it. So describe the types of evidence that can be used to infer. Well, that's what we're talking about here, evolutionary relationship. Okay, so phylogenetic trees and cladograms show evolutionary relationships among lineages. Phylogenetic trees and cladograms both show relationships between lineages, but phylogenetic trees show the amount of change over time, calibrated by fossils or a molecular clock. We'll briefly talk about those. Traits are either gained or lost during evolution can be used to construct phylogenetic trees and cladograms. Okay, shared characters are present in more than one lineage. Shared derived characters indicate common ancestry and are informative of the construction of phylogenetic trees and cladograms. The outgroup, we'll talk about outgroups and ingroups. Molecular data typically provide more accurate evidence, so they look at DNA similarities. And then, phylogenetic trees and cladograms can be used to illustrate speciation that has occurred. The nodes on a tree represent the most recent common ancestor of any 2 groups. And they can be used to, can be constructed from morphological similarities of living or fossil species and from DNA and protein sequence similarities. They represent hypotheses, so these are ways of understanding the world around us. So this kind of falls under systematic biology, basically just kind of trying to understand like the unity and diversity of life and the history of biodiversity. Taxonomy is kind of identifies and names organisms. And so this started a long time ago with Aristotle. And then Carolus Linnaeus was like the father of modern taxonomy, and he came up with like the genus and species. And so every organism on the planet has a genus and a species, name. And so today taxonomists use natural groups based on shared evolutionary history. And so phylogeny is a study of a family tree based on evolution. So these 2 butterflies share a common ancestor, and this is a common ancestor, this is a common ancestor, all the way back. But similarity-wise, these 2 would be very similar to each other. And so, back in like teaching middle school days, King Philip came over for a great spaghetti, or did King Philip come over for great spaghetti? Any of the sort of that stuff. And so, but here's where it kind of comes into play. Like, you have like the biggest, domain is like the largest, most inclusive, and then all the way down until you get to a specific species. So every living thing on the planet that has been named and identified has, everything just has one name, which is the genus and species. All right, so Panthera pardus, there you go. And so the higher the category, the more inclusive, of course, which means like we have tons of, like millions upon millions of organisms here and stuff. And so standardizing nomenclature is difficult, but usually we look at DNA sequences. It's called DNA barcoding, compare short fragments of DNA sequences. And then Carl Woese sequenced, prokaryotes in like the 70s, and he found like prokaryotes weren't all the same. And so this, he proposed that the former Monera kingdom be divided into bacteria and archaea, and that is true today. So there's 3 kingdoms: eukarya, archaea, and bacteria. Bacteria, this is just kind of falling under systematics, something that you might need to review a little bit from biology days before phylogenetic trees are brought up. But bacteria, yeah, of course, heterotrophic, some are photosynthetic. Cell walls contain peptidoglycan, of course, E. coli and tetanus and anthrax. Archaea is very different. They don't contain peptidoglycan. And the plasma membrane contains branched lipids. And so they're very different. They're extremophiles. They live in extreme places. And so, you can see right here the branching is different. You don't need to know anything like that. It just shows you an example. And then, of course, eukarya, protists, and fungi, and plantae, and animalia. Those are our, things there. And so here's the 3 domains of life: bacteria, archaea, and protists, animals, fungi, plants. And so major distinctions among the three domains of life, membrane-bound organelles, yes, eukaryote does have them. Nuclear envelope, yes, eukaryotes have them. Some introns in archaea, but we have introns. Remember what introns are? We don't want those inside the final mRNA transcript. And so systematics, one goal of systematics is to determine phylogeny, which is the evolutionary history of a group. Phylogeny is often represented as a phylogenetic tree. So a tree, tree of life, it's a diagram indicating lines of descent. And of course, we're going to talk a lot about the term common ancestor. Okay, so this is just kind of a big idea. Phylogenetic trees and cladograms are models, so they help us understand the world of common ancestry and evolutionary history that can be used to establish the relatedness of different species through analysis of similarities in morphology, which is how they look, or comparison of DNA and proteins. Of course, morphology would be more in terms of fossils. And so if you look at this, what kind of organism is this? And you're like, but this is silly, right? This is obviously, he's setting me up for something because that's obviously a snake. But it's not. It's actually a legless lizard known as the European glass lizard. So why isn't this glass lizard considered a snake? And then more generally, how do biologists distinguish and categorize the millions of species on Earth, which is kind of where we're at. And so you can see right here, based upon DNA sequencing, snakes lost their, limbs, here. But based upon DNA, glass lizards share more alike with monitor lizards than they do with snakes. And so based upon DNA comparison, it's kind of interesting. So if you want to get ready for this chapter, reviewing those three will be good. And here is some other questions that you can review. Oh, my bad. So right here, sorry, I'm kind of like skipping around all the place, but go ahead and pause it and answer it. And so the answer is C. Over time, evolution by natural selection causes species to accumulate differences from their ancestors. The genome of an organism only includes the organism. Well, that's not true. No, never identical. Includes all, yep, C. And then number 3, suppose that two distantly related species live in similar environments and share a distinctive characteristic that facilitates survival, but it is not found in their immediate ancestors. So this distinctive feature would most likely represents, well, homology would represent common ancestors, so this would be an analogous character. Okay. From another textbook, phylogeny can help us resurrect protein sequences from extinct species. We probably won't dive into that, but that's just kind of interesting. And so phylogeny is the evolutionary history of relationships. And then a phylogenetic tree or a cladogram is a diagrammatic reconstruction of that history. And so here's an example of one. So you can see we have a common ancestor at every node. I'll talk to you about that in a second. Gray wolf and the coyote. You can see how the genus, right? The genus is, and the families, they all belong to the order of Carnivora, which means they eat meat. And so that's an example of phylogenetic tree. So a phylogenetic tree is, sorry, I'm not moving the right thing. Well, I'm still not moving the right thing. It's fine. An ancestor and its descendant populations form a lineage. So species, certain time, and then the lineage goes through time, okay? And then when a single lineage divides in two, so there's some sort of speciation event. We'll talk about that in the last lecture. A split occurs, divides into two descendant lineages, and they continue to evolve independently, represented by red dots, okay? And then you can, continue to split over time and over time. And so the splitting of events is shown by the position. So this could be a speciation event, it could be a gene duplication event, transmission event, and we'll talk about that in our speciation. That's just like a little preview. Each branch point represents a common ancestor. So here's a graphic from another textbook that I really respect and like. So a phylogenetic tree visually represents a hypothesis of how a group of organisms are related. So you can see right here, each horizontal branch represents an evolutionary lineage. Each position along the branch represents an ancestor in the lineage, leading up to the taxon named at the tip. Chimps and humans are an example of sister taxa, which means These are groups of organisms that share a common ancestor that's not shared by any other group, so they're sister taxa. They're very closely related. Each branch point represents a common ancestor of all the animal groups shown. So here's a branch point for everything, but that would include everything except fish. That would include everything except frogs and fish, and then that would include everything except the others, okay? And so this is alternative forms. These diagrams are referred to as trees. You can do a diagonal tree, or you can do a vertical tree. Not really sure what I would recommend. It's kind of preference, guys, but you know. Notice something here, and this is very important because they might trick you on this May 5th. You can rotate like any node, okay? So if you rotate at any node, you can get a lot of different varieties of trees, but they all mean the same. You're like, well, this doesn't look the same, but it means the same because if you look closely on like the X and the common ancestors, they're all the same. It's just rotated, okay? Now, you would probably draw it like this, which we all would, myself included, but sometimes they show it like that just to kind of throw a little wrench in the works. A phylogenetic tree may portray evolutionary history of all life forms, major evolutionary groups, even genes. Okay, and so here is an example of mammals, right? And you know, mammal glands, opposable thumbs, all of that is an example of that. So phylogenetic trees show patterns of descent, not phenotypic similarity. Okay. Understand phylogeny can have practical applications. Considering like corn, right? From a phylogeny of maize based on DNA data, research have identified 2 species of wild grass that may be maize's closest living relatives. So these could be used as reservoirs of beneficial alleles. You know, genetic diversity is a good thing. Phylogeny provides important information about similar characteristics in closely related species. So for example, they've used this in like crime labs and stuff. So sequences by and I'll show you a picture here in a second, but yeah, whale meat. Where did whale meat originate? And so a different use of phylogenetic trees is to infer species identities by analyzing the relatedness of DNA sequences. So whale meat, some whales are legally protected, right? So what happened was a group of scientists, you know, took some whale meat, I believe it was in an Asian country, and you know, these guys are endangered, protected. You can see it, hence the red, like mitochondrial DNA, and that was like super closely related. And so, by all accounts, had to come from these whales that are protected. So that company, of course, got in tons of trouble. So the sequences of 6 of the unknown species were most similar to mitochondrial DNA sequences of whales are not legal to harvest. Vertical distances between branches do not have any meaning, right? So you can rotate them and stuff. So cladistics is a way to analyze primitive and derived characters by the construction of trees, called a cladogram on the basis of shared derived characteristics. So a cladogram is a special type of phylogenetic tree. Okay. A clade, and I'll show you some pictures here, but a clade is an evolutionary branch that includes a common ancestor together with all its descendant species. Again, we're always talking evolutionary history here. So again, or classifies organisms by common descent. So what you have here is a couple, I think I gave you this picture here, but what you have here, each color rectangle represents a clade. A clade is a taxon that consists of all the evolutionary descendants of a common ancestor. So common ancestor, common ancestor, common ancestor, common ancestor. A taxon is any group of species that we designate with a name. So you can see here different types of groups. I wouldn't really know these though, but here you can see right here, mammals, amniotes, tetrapods, vertebrates, getting more and more specific as we go down the tree. And so evolutionary relationships among species form the basis for biological classification. As new species are discovered, these things are reviewed and revised. The tree of life's evolutionary framework allows us to make predictions about lots of different things. So they serve as models of understanding. So each character of an organism evolves from one condition, the ancestral trait, to the derived traits. The derived traits are new. Shared derived traits provide evidence of the common ancestry of a group called synapomorphies. Not sure if you need to know that term. I don't think you do, but it's just nice to know. That's just derived character traits. Okay, so you have a shared one. That's like an ancestor of the taxon, and then a derived one, which is a novelty unique to a particular clade. So a character can be both ancestral and derived depending upon the context. So for example, when considering the entire animal kingdom, the backbone is a shared derived character because it is present only in vertebrates. But when considering only the group including mammals, the backbone is ancestral because it is present in mammals and all other vertebrates that are not in that clade. So it depends upon the context. What are they asking you? What are we doing? So in-group is the group of species being studied, and out-group is close related but doesn't have all those parts. And so an example from a textbook is this. So you can see the taxa here, the names, characters. The land-split is the out-group. It's related, relatively speaking, but it doesn't have any of the characters. Characters is absent. And so you can see the lamprey, vertebral column, the lamprey has that and everything else, but it doesn't have, so the zeros, right? And so if you were to build a phylogenetic tree, based upon this, you would make the land-split the out-group. And again, building one, sometimes they ask you, usually they have it built for you and you label it, or you have to analyze one that they've already made. And so, but yeah, everything, so this is the evolution of that trait. So everything after that, line right here should have a vertebral column, and they do, lamprey all the way through leopard. Hinged jaw is only the 4, forelimbs is only the 3, amnion is turtle, and leopard, and then leopard is the only one that has hair. So this is a phylogenetic tree. You can see right here, here's a tree. Here's the derived characteristics based upon the following clades, labeled like that, and they built it based upon that chart right there. Okay. Branch links can represent genetic change. So this is just comparing sequences of a gene that plays a role in development. Drosophila was used as an outgroup. You can see that the human and mouse is very close. They don't share many differences here. The human shares more differences with the chicken and the frog and the zebrafish and lion and a crazy amount with Drosophila. So it could represent genetic change. It also can represent time. Of course, evolution has to have millions of years to, work out all the mutational challenges. And so similar traits can also develop in unrelated. We talked about this convergent evolution, and we talked about that with the sugar glider and flying squirrel. A trait may be ancestral or derived depending upon the point of reference. Okay, feathers are an ancestral trait for modern birds, but in a phylogeny of all living vertebrates, they're a derived trait because birds are the only ones that have feathers. And so yeah, do a little practice here with me. So you can see right here, shark and dolphin. Think about some similarities. And then predict whether these traits show evidence of synapomorphies, which means traits that are derived relatively recent, and justify the claim with evidence. And well, hopefully you think about, okay, well, the shark is, it's cartilaginous fish. We're not marine here, but like breathing air, not breathing air, bones, cartilage, very different, developments and nursing young and stuff. And so you can see cartilaginous fish are pretty ancient. Aquatic vertebrates evolved this is the vertebrate. And then mammals are right here. Mammals return to the water and evolve the aquatic body layout. So that's convergent evolution because they don't share a recent common ancestor. Clades are often guided by the principle of parsimony, which means the minimum number of assumptions. Simplest is best. So the best cladogram is one in which the fewest number of shared derived characters are left unexplained. So you could draw this multiple different ways. But why don't you go ahead and practice this. So first off, you can see the land-split is the out-group. The in-groups are what we're studying. And you might be like, okay, well, the next branch should be vertebrate, the next branch is forelimbs, and the next branch is probably amniotic egg. But that's where the problem is right here, right? What do we do with mammary glands, and what do we do with gizzards? And so hopefully you see that these 3 are probably in a clade, and these 2 are probably in a clade. And so let's see. And so you can see right here, amniotic egg, and then there's our 3, right? lizard, crocodile, finch, feathers, gizzard, epidermal scales, and then hair, mammary glands. Long canine teeth evolved in the terrier, and a large brain developed in the chimp. Okay, so visual skills. Which of these trees? So go ahead and practice this problem. Which of these trees shown here depicts an evolutionary history different from the other two? And so hopefully you showed that C shows a different evolutionary history. Why? Because C and D are both sister taxa in this phylogenetic tree. The C and B are sister taxa here, so that's completely different lineage. Okay. You can try to draw this. Go ahead and pause it. Try to draw this. Try to see what you get. We'll go over this answer. I'll show you the answer here. Obviously in this, but we'll try to go through some practice problems here. So yeah. All right. So hopefully you did it. And so there's a couple of different choices here. Assume their sister species and redraw the tree accordingly, right? And so when you do that, when the tuna and dolphin are sister species, you have to have multiple evolution points along the line for dolphin 3, 4, 5, 3, 4, 5, right? And so that is not parsimonious. This one's parsimonious. Less explanations, less things needing to happen. The simplest explanation. Okay, so simplest explanation is 6 is a derived character evolving independently in the tuna line versus 6 and then 3, 4, 5 as well in the dolphin. That doesn't make sense. So this is the better tree. Okay, phylogenies are inferred from similarities in morphology, genetics, and biochemistry. So similarities, right? Phylogenetic and genetic similarities due to shared ancestry are called homologies. So organisms with very similar morphologies or DNA sequences are likely to be more closely related. So systematists must distinguish between similarities resulting from homology, which is shared ancestry, right, and analogy, which is convergent evolution, similar traits being selected for in similar environments. And so, for example, an example of this is Australian moles and African gold moles are similar in appearance, but the evidence indicate they do not share a recent common ancestor based upon phylogenetic science. So you can see they look very similar. The likelihood of homology based on complexity also applies to genetic comparisons. Here, it's not a blank, but I would underline this. Let me underline it for you. The more similarity in nucleotide sequence, the more likely the genes are to be homologous, which means share a recent common ancestor. Okay. Molecular homologies are based on the degree of similarity in nucleotide sequence among taxa. Okay, a single nucleotide insertion or deletion shifts the whole sequence, but computers can help us see that, okay? So what you have here is like homologous DNA sequences of species 1 and 2. Deletion and mutation shift what had been matching in the previous one. And then all of a sudden we matching reads realign after a computer program adds gaps. So those portions still align once the length is adjusted. It's very similar. We've just added some stuff that possibly has meaning, possibly does not. All phylogenetic trees represent hypotheses. They're modified when new evidence shows up. And so we can use it, since it's a model, we can use it to predict. For example, characters of dinosaurs are inferred from characters shared by living birds and crocodiles because they, are relatives. There's a common ancestor. Crocodiles are crazy old according to evolution. And so common ancestor of crocodilians, dinosaurs, and birds, right? And so what traits do birds and crocodiles, and so we can kind of start, filling in the gaps there based upon phylogeny. 4-chambered hearts, singing, nest building, brooding behavior. These characters likely evolved in the common ancestor. Likely, of course, we don't really know, but it's a prediction based upon evidence. Fossil discoveries of nests and brooding in dinosaurs support this hypothesis. And there's a crocodile guarding its nest. Don't mess with that. And there's just artist renditions of this. Here's a fossil remains with eggs, kind of like surrounding the eggs, fossil evidence. And so applications are kind of cool because zoonotic diseases are infections. This is not cool, but like applying phylogenetics to helping humans live and treat diseases is cool. And so it is important to understand when and where. So for example, one example, and it's wreaked havoc on our planet, is HIV. And so HIV is a zoonotic disease. It started, in chimpanzees, but one version started in pseudomangabase. And how do we know this? Well, we did a phylogenetic tree. We started comparing, the genomes, and we started seeing, okay, there's a lot of SIV, which is simian immunodeficiency virus, and not humans, infecting other primates. And so you can see right here, there's a lot of different viruses, right? The HIV-2 and HIV-1 are more related to the ones that came from pseudomangabase and chimps. And so forensic investigations also, for example, a physician was accused of injecting blood from an HIV-positive patient to his former girlfriend in an attempt to kill her. That's tragic. Phylogenetic analysis revealed that the HIV strains present in the girlfriend were a subset of those present in the physician's patients, which means they have way more in common than they should have with his patient. And then here's viral isolates from other HIV positive in the local community. So phylogeny, man's behind bars. Molecular clocks are an interesting thing as we close out this lesson. And so a molecular clock uses the average rate at which a given gene or protein accumulates changes to gauge the time of divergence. So what's the average rate we see? And then we use that as kind of, what's the term? Uniformitarian, in a uniformitarian way, which means the rate that we see today has happened throughout life. And so they must be calibrated using independent data. Okay, so here's mutation rates. So the slope represents the average rate of change in amino acids based upon what we have observed, right? So in order to see a certain amount of amino acid change, we're talking millions of years for that to come into being. Okay, so molecular clocks help with this, help us understand, and also dating, radioactive dating and stuff, which we'll see here in a second. So a molecular clock was used to estimate the time when HIV-1 first entered human populations. It was calibrated using biopsy samples in the 80s and 90s, and then tested using samples from the 50s. So they're looking for mutations, right? And so what they did was they saw, okay, here's the samples, confidence, that's all statistical analysis. And then roughly, maybe around 1930 was the HIV-1, based upon mutation rates that, okay, mutation rates happen between these 2 years at a certain rate. Let's bring that all the way back to the beginning. And so application of molecular clocks suggests by 1930. So, all right, so here's a practice problem that I put for you in your notes. Good FRQ practice problems. Please do this before class. We'll talk about it. We'll practice it. I'll probably have one more FRQ practice problem or some sort of other problems for you, maybe biozone. But it's been a great time learning about phylogenetic trees. Hopefully, it's been a great time for me. I mean, making videos that don't have really cool effects. I'm trying to work on it. I'm just wasting time right now, so I just need to let you go. But I hope you are doing well when you watch this. Again, we've got not much time left, right, before that test. And so let's give our all. Remember, you are working for, lots of money, lots of time in college. It'd be so great not to have to take courses if you don't have to. So you are so smart and so kind, and you're working so hard. That does not escape my notice. May God bless you, give you peace in your work. I'll see you soon.